Good evening, friends. Uh, Today I am dealing with the topic cell signaling and its role in oncology. And basically, this topic is a continuity of our first video, that is the concept of molecular oncology. For those uh, oncologists practicing the targeted therapy, the four chapters are very vital. That first is I've already delivered, the second I'm going to deliver today, and the third is a concept of angiogenesis, and a fourth is the concept of apoptosis. So myself, Major Dr. M. Beg, on my YouTube channel, the Major Beg Oncology classes. Today I am dealing with the cell signaling and its role in oncology. So the modern principles of carcinogenesis involve the multi-stage process in the development of a cancer. And basically the cell's correct functioning entirely depend upon the signaling to the cell. If a bad signal, the cells will alter its behavior. And then a proper function of the cell signaling pathway is responsible basically for a cellular growth, cell differentiation, apoptosis, angiogenesis, metastasis, all is related to the correct signal a correct signal or a indirect, if a bad signal. So it's entirely depend upon these factors. So what cell signaling deal with? Basically understanding the receptor, overexpression for signaling pathway for cancer, new possibilities of target therapy. If you discover a new receptors, you anti-receptors you can design, and then by the genetic engineering, and then you can impose the target therapy. Alteration happen in signaling pathway, and how it alters, we will be discussing it. Oncogene overexpression and their influence on the signaling pathway in the cancer, and finally deconstructing the signaling pathway and optimizing the targeted therapy for a specific cancer. So that is all about the cell signaling deal with. The cell sends the signal from extracellular domain and intracellular environment as well as and directly from the other nearing cells. So they have any cells having a three kind of a signals: one within the cell, or one out of the cell, that is the extra cellular matrix, and then extracellular environment as well as the neighboring cells. Cell respond to these signals in a variety of ways, like primarily modifying the protein level and protein level of production and its activities, and the protein levels are controlled by the rate of transcription, translation, and proteolysis. So these all are interlinked. So what is all about the transcription? You must have heard about many times. The transcription in a biology is the process by which the cells make an RNA copy of a piece of a DNA. This RNA copy is called mRNA, messenger RNA. It carries the genetic information needed to make a protein in a cells. Basically, the mRNA, messenger RNA, goes inside the DNA, and between the two chromatids of a chromosome, it takes the coding and comes out, and then it starts the protein synthesis as a translation, so that a specific protein is being formed according to the messenger RNA, the what kind of a coding he has taken from the nuclear level. So it carries the information from DNA in the nucleus of the cells to the cytoplasm where proteins are made. So this is all about transcription. What is translation? The molecular biology and genetics, translation is a process by which ribosome in the cytoplasm or endoplasmic reticulum synthesizes a protein after the process of transcription of DNA to RNA in the cell's nucleus. So what happens here? The mRNA has taken a coding from the nucleus by the through the DNA and comes out the cell and goes to the ribosome, and where the tRNA attracts And by the help of this tRNA, the coding is transferred from mRNA to tRNA, ribosomal RNA, tRNA, and then a particular protein amino acids are synthesized and a proteins are synthesized of their what kind of a instructions was received from the nucleus. Then what is the transduction? The transduction is different process, is basically process by which the virus transfers its genome, a genetic material, from one bacterium to another, and these viruses called bacteriophages are able to infect bacterial cells and use them as a host to make over the more viruses. So basically the genome is transferred and then the transduction of the genome to the virus infecting bacteria, that is bacteriophage, and that is called transduction. So now the signal transduction, the process by which binding of an extracellular receptors to the cell surface receptors happen. Out of the first step result in the change in the biochemistry, cellular biology, genetic transcription happens. I mean, because of this, example, the cytoskeleton protein Merlin, that is neurofibromine 2, a tumor suppressor gene. If it is lost, it causes the increased proliferation of a tumor cells of neurofibromatosis type 2. So this is important. The signal transduction pathway regulates the differentiation, division, death in the mature and developing cells. Some pathways are common for all cells, but some pathways are very specific, specialized cells. For example, like you see, the synthesis and secretion of insulin not happens by any other part of a tissue in the body except the pancreas. And a migration, and further, like you see, migration and phagocytosis by the neutrophils is also a very unique process. Specific, likewise, the abnormal behavior of altered cells, that is cancer cells, start uncontrolled division and invasion and a further gone to the seedlings to the other side, that is metastasis. So these are because of the signaling. So the common issue faced by the cell in the signal transduction is two. One, how is the signal sensed, and how are the level of activities and the level of protein modifying the response to the signal sensed. That is the two important point in the sense signaling. The most signals are initiated by the ligands and are sensed by the receptor to which they bind. Binding of ligand to the receptor stimulates the activation of a protein necessary to continue the transmission of signal. Though the formation of multi-protein complex and a generation of small molecules, that is known as the second messenger, develops. So what is second messenger? So in that way, I'll take up in the sensory machinery, ligands and a receptor, the signals transduction molecule, that is the second messenger that initiate the signaling cascade, including the proteins, nucleotide, lipid, amino acids. These all can be a second messengers. The most extracellular signals, which are protein, bind to the receptors present on the plasma membrane of the cells. I mean, most of these extracellular signals are a protein, binds to the receptor present on the plasma membrane. There's the other molecule, especially those are steroid. They directly diffuse inside the cell and bind to the receptor in the cytoplasm and finally reach to the nucleus and act there. So these the steroid hormones, they behave like this. The signaling molecules originate from variety of a source either outside of the cell or within the cells. Also responds to the signals that arise from within. For example, checkpoint pathway is that ensure progression of cell cycle. A cell cycle which also sense DNA damage and do necessary repair before the next mitosis happens. So that is the cell's immune checkpoint inhibitors, which I am trying to indicate you. So let us talk about some few portion of the receptors. The plasma membrane of a cell serves to insulate the cells from outside the environment. But the barrier must be breached to transmit the signals from extracellular origin across the plasma membrane. And this can happen by two, either by activation of transmembrane receptors or by using a ligand that are membrane permeable and can go to the cross the plasma membrane. The amplification of most of the signal is necessary for cellular response. Receptors bind and respond to more than one ligand. Usually the receptor bind with the one ligand, but sometimes one receptor bind to the more than one ligand. Like example, the epidermal growth factor receptor bind to two ligands. One is transforming growth factor alpha and second is a heparin binding factor beta. The activation of a receptor is a lead to stimulation of several downstream pathways that stimulate the different targets. The receptor also activate other receptors like activation of epithelial growth factor receptor by G protein, coupled receptors that is GPCR, which occur as a result of protease cleavage and activation of heparin binding epidermal growth factor. So there are two important factors. The tyrosine, insofar in the oncology is concerned, the tyrosine kinase is a very unique receptor. Let us embark upon this receptor. The receptor tyrosine kinase are a subclass of a tyrosine kinase, multiple kinase, that are involved in the mediating to cell communication, controlling a wide range of complex biological function of the cells, metabolism of the cells, proliferation of the cells. These functions are cellular growth, motility, cellular differentiation, metabolism. The receptor tyrosine kinase are high affinity cell surface receptor for many polypeptide, growth factors, cytokinins, and hormone. So till late, almost 90 unique tyrosine kinase genes identified in the human genome. Out of that, the 58 encode receptor tyrosine kinase proteins, which is very important so far as the oncology is concerned. Thus, any alteration in tyrosine kinase receptors are usually associated with the development of a cancer, and their activity can be manipulated by targeted therapy. The targeted therapies inhibit signaling pathway, resulting in an arrest of cell growth, apoptosis, additionally bind external receptors with a mediate antibody-dependent cellular cytotoxicity. So you see here the tyrosine kinase. I told you what are the several actions that tyrosine kinase does. Here in this figure you see the tyrosine kinase receptors is combining with the multiple receptors. Can combine epidermal growth factor receptor, vascular endothelial growth factor, platelet derived growth factor, ABL gene. So after combining with these receptors, it comes here cross the plasma membrane and lead to the several signaling pathway. There are three most notable pathway are MAPK, that is mitogen activated protein kinase. So what is mitogen? Mitogen is basically a kind of a protein that induces cell division and a mitogenesis. So they ignite this pathway. And the second JAK-STAT pathway, that is Janus kinase enzyme, and signal transducer of activated transcription, that is STAT pathway. And the third is the PI3K AKT pathway, that is the phosphatidyl inositol 3 kinase pathway, or AKT alpha serine threonine protein kinase pathway. So these pathway, if keep on going, they will lead to metastasis, they will lead to cellular proliferation, they will lead to angiogenesis, tumor progression. So what does TKI does? It stops this tyrosine kinase inhibition, and this tyrosine kinase inhibition lead to stoppage of these signaling pathway and checks the see, you know the very famous drug as the imatinib, is in chronic myeloid leukemia, that is the ABL gene through the TKI, that's a TKI, the tyrosine kinase inhibition. We can check this chronic, have a very good response with the in chronic myeloid leukemia. And there are several drugs in the developed as a tyrosine kinase inhibitor. So the receptor tyrosine kinase are the transmembrane protein that have an extracellular ligand binding domain. The transmembranal domain and a cytoplasmic tyrosine kinase domain. Ligand for these receptors is a protein or polypeptides or peptides. Activation of receptor tyrosine kinase generally require a tyrosine phosphorylation. That is also tyrosine phosphorylation is an important step in the progression of the signaling. So tyrosine kinase, what action is there? Receptor that activate tyrosine kinase receptor. I have already explained in the previous diagram how the tyrosine kinase act. Tyrosine do have an intrinsic enzyme activity which stimulate associated factors of tyrosine kinase. The example of this kind of receptors include cytokinin, interferon, cytokine. These are there. What is dimerization? This term is coming again and again. That the binding of a signal molecule to extracellular domain of a receptor tyrosine kinase molecule cause two receptor molecule to dimerize. What is actually this? Dimerization is basically a chemical reaction that joins two molecular subunits resulting in the formation of a single dimer. That is called dimerization. This brings the cytoplasmic tail of a receptor close to each other and cause tyrosine kinase activity of these tails to be turned on. You see here, in this figure, both the receptors are close together, inactive receptor tyrosine kinase and you have active receptor tyrosine kinase. Here in this an active portion, you see here the two uh, molecules are close together and dimerization happens and lead to the mitogen activating pathway, the PIK3 pathway, AKT pathway activation, and that lead to the cellular progression and mitogenesis and lead to the angiogenesis and also on tumor progression. So tumor necrosis factor is another factor we should stress upon. The tumor necrosis factor is an multifunctional cytokinin that plays an important role in the diverse cellular events such as cell survival, cell proliferation, cell differentiation, and death. As a pro-inflammatory cytokinin, basically the tumor necrosis factor is a cytokinin released by the macrophages, inflammatory cells. So the tumor necrosis factor is secreted by the inflammatory cells which may be involved in inflammation associated carcinogenesis. So you see here the tumor necrosis factor inhibition may lead to the cellular progression, tumor progression. So the tumor necrosis factors are cytokinins of small molecules that modify the response of human body to external signals. And tumor necrosis factor was found in 1975, way back 1975. Its main activity was assumed to be a lethal for cancer cells and to be able to kill the tumors, hence the name given the tumor necrosis factor. The tumor necrosis factor receptor is undergo oligomerization after the ligand's binding, which is necessary for signaling stimulation of a receptor, lead to the recruitment of cytoplasmic protein that binds to each other and the receptor through the death domain. So what is actually death domain is a homotypic protein molecule composed of bundle of a 6 alpha helix is related to sequence and a structure and ultimately form the capsase recruitment. The death domain is found in many proteins that are involved in signaling pathway related to cell death and thereby activating protein cascade 8 that initiate the apoptosis. So that is the basically caused by the tumor necrosis factor. Here you see the macrophages releasing the alpha tumor necrosis factor, and this tumor necrosis factor lead to apoptosis and lead to apoptosis, so that any change in a receptor status of a tumor necrosis factor alteration lead to the proliferation of the cells and a proliferation of a tumor. What is nuclear receptor? Lesions for nuclear receptor diffuse in the cells and bind the receptor present either in the cytoplasm or in the nucleus. The legend include the steroid and retinoids, thyroid hormone. They directly goes up to the nucleus and hit DNA and an action starts. It does not go work on a membrane, it directly go to the nuclear level. The propagation of signal to the cell interior, how it happens. Though the mechanism of various receptor and ligands that initiate cell signaling are very different, but the most receptor activate a set of common downstream molecules to transmit their signals. The molecule that transmits signals are basically what is protein, lipid kinase, GTPase, phospholipase, protease, adenyl cyclase. These are basically molecules which transmit the signals. These molecules produce array of response, including the change in the transcription, translation, enzymatic activities, proteins formation, and cell motility. So the regulation of protein kinase, the balance between the protein kinase and the phosphatase activity, control the protein phosphorylation. Protein kinase themselves transcription factor and a cytoskeletal components are a few examples of protein regulated by phosphorylation. So phosphorylation is a very, very important factor in the regulation of protein kinase enzyme. Most single transduction pathway require protein tyrosine kinase, a receptor that are not themselves tyrosine kinase. Usually, the several cytoplasmic tyrosine kinase, including the SRC, SYK, JAK family, the phosphorylation of protein and tyrosine can either stimulate or inhibit the enzyme activity. So, regulation of phosphatase, the protein phosphatase remove the phosphate residue from protein, which can either activate or inhibit the signaling pathway. Like GTP, that guanine triphosphate binding protein, the protein instruction is also important mechanism of signal transduction. The G protein which binds with GTP are best studied protein till now. The mediators that regulate the other protein by direct binding result in conformal changes. The GTP binding protein act as a digital switch. The second messengers, what are the second messengers? They are the intracellular signaling molecule released by the cell in response to exposure to the extracellular signaling molecules. Because of the first messengers, the first messenger, the process lead to the development of a second messenger. The second messenger triggers the physiological changes at cellular level, such as proliferation, differentiation, migration, survival, apoptosis, and depolarization. The small molecule of a second messengers, the small molecule transmits signals by binding non-covalently to the protein target and affecting their functions. These molecules cause second messengers, such as growth factors binding to the cell surface receptor, is basically a second messenger. The cyclic AMP use the first second messenger discover adenyl cyclase activated by G protein catalyzes and synthesis of cyclic AMP protein from ATP. The primary target of cyclic AMP is a protein kinase. The second messenger and the calcium relation in the cell cytoplasm. The second messenger adenyl cyclase activated by G protein, it catalyzes the synthesis of cyclic AMP protein from ATP. These cyclic AMP primarily target protein kinase, which binds with the calcium channel in endoplasmic reticulum and stimulate the release of calcium in the cytoplasm of the cells. So above action lead to the influx of a calcium from extracellular space to inside the cells through the plasma membrane, which lead to the calcium flood in the cytoplasm, and then that lead to the this calcium flood lead to the enhance the sin signaling process, And calcium inside the cells lead directly regulatory action of enzymatic activities, that means signaling activities, ion channel transcription activities, and lead to the cell signaling carry on to execute their effect at the cellular level. Thank you very much, guys. This is a very simplified version of a cell signaling which I could have served you before you all, because as such a simplest form you won't be able to read out from any book. So you have to go one by one, two, three times of viewing of this uh, lecture so that you can understand. And basically, the tyrosine kinase is a very important receptor which we need to understand because involved in the various cancers, and there are these cancers and then targeted therapies you are practicing. You should know these things. Thank you very much. Stay connected, and I also request you all guys to help your other friends to subscribe this channel in order to enhance their knowledge. And spreading of a knowledge and a light is always you. It's like something. It's a God blessing basically. Thank you very much.